Hey friends, like we were discussing about the protein folding in the last class. So in today's class, we'll be discussing about the impact on how fast a protein can fold. And this actually covers the paradox, which is also called a Levinthal's paradox. Levinthal's paradox says, so let's suppose if we have like n number of amino acids, right? So the possibility of assessing each one of those towards a particular structure is always ambiguous. So what does this mean is, so for example, if you have like 10 amino acid, and if you think these 10 amino acid is going to uh, form a particular structure. So for example, if these are the 10 amino acid and these are going to form a globular structure like this, right? So instead of going toward this particular structure, they can also form a fibrous sort of protein or some different shape of protein, right? So this is called Levinthal's paradox. And how exactly this happens, nobody knows, but there have been hypotheses. But how is it relevant towards the IIT exam or the GATB exam is because, so let's suppose this is the graph, right? In this graph there's energy, and here is the native conformation of the protein. So what is the native conformation is where protein is actually functional. Okay, so for example, we have like a random structure of our protein, right? So here it is attaining the highest energy. To be in that particular situation, protein must be in like very high sort of energy. So meaning system should get the energy from the surroundings. Oh, I do not like this pen. Okay, so system should get the energy from the surrounding. So I hope you must know about the thermodynamics, right? So this is system, and so this is system, and this is surrounding. So mass and energy transfer can take place, due to which what happens is, so if it does not get like enough energy, so the main goal of the protein is to come to lowest possible energy. Lowest possible energy. Due to which what happens is it can actually sustain in that particular conformation and can remain functional active. Okay, so for example, if this is a protein, it can form a, let's suppose, coil structure or some sort of beta sheet structure, right? And it can eventually attain some sort of either the globular structure or the fibrous structure, which are going to be functional, and these protein will have lowest possible energy. Okay, so main goal of any protein folding is to bring a particular protein to a lowest possible energy. Okay, so now what are the things that can actually affect this protein folding? So like we discussed, so from primary structure it goes to secondary, from secondary it goes to either tertiary, and from tertiary it can go to the quaternary structure, right? So mostly our protein is limited in usually in the tertiary structure, but like some of other protein can come and make it a functional protein and can go towards this quaternary structure, right? So there are bondings that take place, right? So there is hydrogen bonding, there are non-covalent interactions, which include hydrophobic interaction, dipole-dipole interaction, or dipole-induced dipole, and van der Waals, right? And also there are some sort of covalent interactions, and this happen in cysteine, like we have discussed, right? So when two cysteine molecule join together, cysteine, they form something called cysteine. Okay, here is E. Okay, and it has group like this. Okay, so let's suppose this is one amino acid, this is another amino acid, cysteine amino acid, and this bond is called disulfide bond. Disulfide bond, and due to which two cysteine are also called cysteine, right? So when you add beta mercapta ethanol, also written as 2 mercapta ethanol, it actually reduces these sulfur bonds, and it disrupt the overall structure. It disrupt the overall structure. So when you put acid or base, like very strong acid or base, so you are actually fluctuating the pH of the system. Due to the fluctuation of pH, the hydrogen bonding, hydrophobic interactions, as well as van der Waal and dipole induced dipole and dipole dipole interaction are going to get weak, and protein will lose the conformation. Protein will lose the structure. Structure or conformation are the same thing, just remember that. Okay, also heat. So heating a protein at like 90 degrees or more than that, let's suppose 100 degree, will denature the protein, and this is the condition where most of the protein get precipitated and cannot reverse. So for other instances, so for example, if I remove the acid or the base, right, so protein will get back to its original structure, right? Protein will get back to original structure. So that is called native conformation. Same here, if I remove the beta mercapta ethanol from the system, these cysteine are going to come together and they are going to form this disulfide bond, and we are going to get the native conformation again. But in case of heat, this process is irreversible. So there are other factors also, such as environmental factors are there, physical pressure is there, which can actually disrupt the protein. But in general scenario, in IIT JAM as well as GATB, these questions are asked in these two formats only. So for example, if you add too much of strong acid or if you add too much of strong base, what happened to the protein structure? Will it be denatured? Will it remain in the same scenario, or like some of the amino acid will get like protonated or deprotonated, stuff like that. Okay. Now we have discussed about the structure of the protein. Now we have also covered the factor which affect the structure of the protein. But one thing that we have forgot is, how do we study the actual structure of the protein? So basically there are only two ways, not two ways, but majorly it is usually through the NMR spectroscopy, and second is X-ray diffraction. Okay, so where we make crystals and we diffract the light, and based on that we assess the atoms, and based on that we get the particular structure. So these are the two ways through which we actually study the actual structure of protein. There is third way called cryo electron microscopy. Okay, so I'm writing it as a cryo, which also detect the crystals, and based on that only we assess the individual atoms, and based on that atom we create the actual structure and we study it. Okay, so I hope you got the idea, and this question is actually important because question directly come from here, and as well as like what are the factor which affect the structure of protein. And I hope you got the idea. If not, do watch the lecture again and do comment next lecture so that I can post the next lecture very soon, because it gives me the boost that you guys are actually watching the lecture. Thank you.